All right, why don't we go ahead and get started with session 3 today, our immune session, which will also be chaired by Rong Tian. So Rong, over to you.
Okay, hello again, everybody. And seems like I will never go away. And so this session is immune is the main theme. And unfortunately, our previous planned moderator cannot make it. So I'm sort of stepping as a pseudo-immunologist, and I'm sure the panelists will help me to have this discussion. So we have 3 speakers in this session. We'll start with Luke O'Neill from Trinity College, Dublin, Ireland. Luke, please.
Great, thank you, Rong. Hope you can see my screen. Very happy to take part. Big thanks to the organizer. I'm honored actually to take part in this. It's a great topic. The last session was fantastic. I'm going to be saying similar things, by the way, to the previous speakers. But a big thanks to Bamsi, Jennifer, and Janine for inviting me. I'm going to give you kind of where we're at with immunometabolism initially. And then I'm going to give you 1 or 2 examples and then give you my list of challenges and questions that we would have in the area. And it is remarkable. I mean, I guess other speakers have said this explosion of knowledge about metabolism in the immune system. And this is a thing I did a couple of years ago, actually, on the number of papers with immunometabolism in the title. And look what happened beginning in 2010, and now a massive rocket. I mean, the last year, even more, so it's incredible. The second example, in my opinion, of how metabolism is back with a vengeance is this. There was a very good review a few years ago on the rise of mitochondria, and they had this great graph where they looked number of publications on different organelles. And look what happened. The nucleus began to go down and the mitochondria went up massively. Sadly, the pearl Golgi, don't work on Golgi, they're way down here. But this illustrates how fantastically interesting it's become. And so many papers on mitochondria now is staggering. One reason is in the immune metabolism area, because there's many papers looking at mitochondria in the immune process, for example, and of course in cancer and all kinds of things. And what it means is, of course, metabolism trumps genetics. There you have it, because this explosion of knowledge has overtaken the obsession with genetics. And really what it's about in some ways, if you're an immunologist, and it's unusual, of course, because many people became immunologists to avoid biochemistry and metabolism. But the fact is, many are getting into it now because you get metabolic reprogramming in response to, say, immune stimuli of all kinds. And that metabolic reprogramming will change the phenotype of the cell, of the immune cell. And you might get a change, say, in inflammatory gene expression. And of course, many immunologists are translational. Will that tie into autoimmunity or autoinflammation? So suddenly metabolic reprogramming has become a huge focus for many labs trying to understand how an immune cell might change and differentiate and become activated. And then secondly, how that might go wrong in disease. And this slide here I made about 1.5 years ago. The last time I went to an in-person conference, it was a Keystone conference in Boulder, Colorado, on immunometabolism. And Nav Chandel, you may have heard of Nav, he's a big advocate of mitochondrial research, of course. He had a great phrase: metabolism is the driver, genes are the passenger, was the concept. And one example is variation in genes. In other words, genetic variants do not explain the increase incidence in many diseases. And a good example is asthma allergy. That's gone through the roof. It can't be explained on genetic change. What has changed, of course, is the environment and our behavior. And of course, metabolism is poised to sense these modifications, changing gene expression and causing disease. Understanding that aberrant metabolism may provide new therapeutic targets is the overall idea here. And of course, you all know this in spades as well. The environment will be driving metabolic change, of course. That's tied into the immune system. It's also tied into the microbiome, which is another very interesting area, by the way. I think Mark mentioned bacteria in the last session, and that's extremely important here. There's crosstalk with those, of course there is. And then, of course, what is the environment? It's exercise and nutrition in the broadest sense. There's many other elements in here as well, but those two kind of capture the environmental change. And then the last piece that's happened in the past 10 years is the inflammatory process, which is a very important part of your immune system, is actually causing a change intracellularly in metabolic pathways and giving rise to this reprogramming. So this kind of area has emerged in the past few years, with the environmental influence being really, really important ultimately for the immune response, and it might tie into aberrations in that response. And again, this was mentioned in the last session as well. My passion for a long time as an immunologist was signal transduction. And I worked on toll-like receptors for a long time. Early on, we get into them. And that was my main thing, figuring out signaling pathways. What's really happened is metabolites are tied into signaling, and they will change gene expression. And then we get these effector responses being triggered. And this concept of metabolic reprogramming became more and more evident. In my lab back in 2013 now, I guess, we could show that inflammatory stimuli or danger stimuli would drive HIF, and HIF would drive glycolysis. Whereas if you have things like helminths, parasitic infections, amazingly, they mainly drive OXPHOS. And there was a real difference there between the two outcomes then in these pathways. And of course, metabolism in the 20th century had revealed metabolic reprogramming in many ways, where normoxia, you'll have OXPHOS, hypoxia drove HIF, and that would drive glycolysis very clearly. And you all know Otto Warburg's work on the so-called Warburg effect, questioned recently, of course, by Ralph and other people, but tumors have an aberrant metabolism as well. So the notion of metabolic reprogramming began to enter the field of immunology, and then that began to provide new information on what might be happening in these different situations. And this slide, I'm sure, will interest you as well. And it summarizes 10 years research in many labs. It's not true in every context. Let me give you a rider on this one. But by and large, it looks as if like the inflammatory cells, and that might be so-called M1 macrophages or Th17 cells, they're inclined to be anabolic. And of course, mTOR is the key driver of that, as I presume you all know. Whereas the anti-inflammatory side was catabolic. They seem to have a lot more fatty acid oxidation going on, and you get more OXPHOS in the anti-inflammatory cells. And that seems to be holding up as an idea. And AMP kinase is very important for anti-inflammatory processes. Autophagy is very important as well, and that's tied into catabolism. So again, this division of labor seems to be emerging depending on the phenotype of the immune cell. And one example, and this came up in the last session as well, how pharmacological intervention can be useful. And this is something we found about 4 years ago now. If you drive macrophages, our favorite cell type, with lipopolysaccharide, LPS, as a bog standard macrophage activator, it's from gram-negative bacteria, you get loads of a cytokine called IL-1 being made. If you block SDH in that macrophage with dimethyl malonate, they stop making IL-1, but look at this, they start to make IL-10. Now it's the same cell here, and all you've done is put an SDH inhibitor in. And IL-10 is a very important anti-inflammatory cytokine, and IL-1 is pro-inflammatory. So here we have a shift in the phenotype of that cell by targeting SDH. Who would have thought targeting a Krebs cycle enzyme would alter the cytokine profile from that cell that could be very important for inflammation or an anti-inflammatory event. And again, that notion of reprogramming, which can be reset then through targeting a specific metabolic process. And of course, mitochondria, as I mentioned, front and center, many papers on this. I mean, mitochondrial DNA is sensed by C-gas, TLR9, of course, with cytochrome C driving apoptosis. NLRP3 is the inflammasome, sensing mitochondrial ROS. Loads of examples of quite complex immune signaling pathways coming off mitochondrial products, and that became a real theme. A good example is this, and our lab saw this early on, and so did many labs. Mitochondria go under this massive fission event if you activate them with lipopolysaccharide in macrophages. And again, these are nice fused mitochondria. They completely change in response to a gram-negative bacterial product and become fizzed. And that's a very aberrant response in many ways and can become pathologic. And then just briefly in my own lab, we worked on Krebs then in macrophages, and we did lots of metabolomics and multiomics and various things. We found a key role for succinate as an inflammatory signal, and a second metabolite, itaconate, we found that was an anti-inflammatory signal coming off Krebs. And this is one little piece of data for you. This is the metabolomic analysis of LPS activated human macrophages. Itaconate was an abundant metabolite in those macrophages. To our surprise, we'd never even heard of this metabolite to be bumped into it in this way. What does itaconate do? It's anti-inflammatory in the broadest sense. And in fact, it's being measured now as a biomarker in many situations. Here's our disease that we're all sick of, I imagine, COVID. Look at this, there was a decrease in itaconate, was reduced in COVID severity. Now remember, it's an anti-inflammatory metabolite. So it goes down in COVID, and that might be why you see some of the inflammatory pathology. And what we found when we came across this, it's about 3 years ago now, I guess, it's a very potent cysteine modifier, itaconate. And it can modify cysteines on target proteins, so can fumarate, as I guess you all might know. And there are proto-drugs or drugs in the clinic coming off these metabolites. Dimethyl fumarate is a very effective anti-inflammatory molecule used in MS, for example. And we've made a drug-like molecule called 4-octyl itaconate, based on itaconate, which again modifies cysteines. And that leads me to this. I think a really important part of metabolic output is covalent modification. And someone's mentioned this as well earlier. We know the whole list of them through the history of this, you know, phosphorylation, acetylation, they were the first ones found. Look at all these modifications, malonylation, even dopamine can modify proteins, so can lactylation. And we found, and by the way, of course, this gives rise to a major change in the proteome. Like the genome is simple compared to this. There's a million options here through covalent modification. You've only 100,000 transcripts in cells. And we found itaconate dicarboxypropylate cysteines, an alkylation reaction, adding to this big long list of covalent modifications that are happening. And then, you know, very recently, then our proto drug, as I call it, for octylitaconate, that's been tested in all these models of inflammation. And guess what? It's anti-inflammatory. And this might be sepsis, it could be lupus, psoriasis, COVID, amazingly, in a model of COVID, it's been shown to block there, even in ischemia in various ways. So in other words, this is a metabolite based on a Krebs cycle intermediate, coming off Krebs, as you may know, itaconate and furfuconate. And now we're modulating these inflammatory pathways by using this metabolite. An interesting, ultimately possibly translatable concept, I guess, is the way to think of it. And really then that leads me to my final slide as time is running out. So, you know, this is what's in my mind for this 4D physiology session, I guess, that we're in. What do we need? And of course, again, to say it for the 5th time, the previous session touched on many of these things. This is the very, very complex system. The genome is simple. The genome and the transcriptome is for children. This is for grown-ups. It's intensely complex. I believe Janelia sells itself in a place that addresses complexity. So let's start with that. So many metabolites. I mean, in that metabolomic screen we did you know we could find 80,000 metabolites in that metabolomic analysis of an activated macrophage? And of course, this has to mean stuff outside bioenergetics, clearly. Very dynamic, all about ratios and fluxes, and that's a real challenge for us. I wrote a review last year with Mike Murphy, who many people know. I know Vamsi knows him very well. For immunologists, we said it's not about the metabolites, it's the ratios, you know. It's AMP, ATP, it's NAD, NADH. I mean, this is what really counts. And also the fluxes through these pathways has to be determining. And people, that's in its infancy in sort of integrating that into understanding what's going on here. Number 3, metabolites are like cytokines. They're as potent as cytokines, which are the key controllers of the immune system. And then metabolism drives the phenotype of the cell. That has to be tied into epigenetics and covalent modification, and non-genetic aspects become really to the fore here to explain disease, very important. And this covalent business, I think, is really important because intense complexity to get our heads around there. Mitochondria, as you all know, critical. Why is that? Huge focus. We've got to think about evolution here. I call this a breakdown in endosymbiosis. In fact, my previous slide, I forgot to say it. Very brief, let me go back because I'm proud of this statement. You know, it's basically, you know, endosymbiosis is broken down here. I can't read it myself, but that notion of the mitochondria going rogue, if you will. They are bacteria, remembered, inside our cells. And now they go rogue. I mean, you get a breakdown, you know, in this endosymbiosis. The enemy within reveals itself, and that's got to be a key concept for us, I think. And then we desperately need new technologies. Now, how we invent them, and again, what, you know, how we run them is beyond my pay grade. But we've got to get a better way to measure metabolites, especially in the immune system, in a much more robust way, because immunologists don't like the weight mass spec. So better methods to measure metabolite concentrations. Multiomics is key. And you can combine gene expression change with metabolic change is important. Old ideas on flux and rate limiting steps need revisiting, because that's the old fashioned way to look at it. Dynamic imaging of mitochondria and organelles. More and more on that, please. Let's get more techniques around those. And if we apply this technology, we'll get a better handle on immunometabolism and how metabolic change is governing so much about the immune system. And that's where I think we should be focusing our attention. So I'll finish there, and thank you very much. Thanks, Rong.
You're muted, Ron.
Sorry, thank you, Luke. And the questions from audience, please type into the box. And maybe I can start it first. Look, you know, the immune cells are extremely heterogeneous, and the immunologists really benefited and drive the FACS technology, and they can give you millions of subpopulations of immune cells. And now with the single cell resolution, it's even more sophisticated. And so does the metabolism change follow the same way? I mean,
I think so, Rong. It wouldn't surprise me, but you could use metabolism to type different immune cells even, you know. So we could now use metabolic profiles to identify, say, an inflammatory macrophage versus another type of macrophage, for instance. And the huge multiplicity of T cell subtypes. So Th17s have a very specific metabolic profile compared to Tregs, and they're two of the big cells that we all work on, you know. So I think it's as specific as that. You know, there will be commonalities, of course, between these immune subtypes, but I bet you there'll be really specific metabolic change. Now, the question then becomes, how determining is that for the phenotype of the cell? And it is absolutely determining, because we know we can intervene pharmacologically, alter that phenotype, you see. So it's a really important aspect. Now, the question is how many subtype? We could be there forever. We're trying to figure out the ones we know, basically, you know, but there could be real subtleties between subtypes of each immune cell as well.
So we get a lot of hands from panelists. I start with Vamsi and Jennifer, and be really brave.
You know, Luke, thanks for that talk. You know, during the course of the day already, we've spoken about the importance of having a manipulable system of some sort, right? Everybody loves in vivo, of course, and then we all acknowledge the limitations of pure cell culture. And we've heard about the heart, the liver, some musings about islet cells, even simpler systems. But I guess I have a question about the immune system in particular. I mean, it seems to me that that may be special in the sense that you can actually do blood draws or bone marrow harvest. You may be able to study them ex vivo, and you may even be able to, you know, reconstitute bone marrow as well. And can you give us a sense as to whether the immune, I mean, to what extent can you study sort of mixed populations of cells, communities of immune cells ex vivo? I mean, is it, again, I know that it's not as good as in vivo, so that's not my question, but is it reasonably good?
You can, Vamps. You can use whole blood, for instance, amazingly, even as an experimental method. It's a bit like organoids in a way, I suppose, but you can certainly take ex vivo immune cell types. You can take them in co-culture, for example, other cell types. So it's feasible. There's always a criticism of the media, of course, as you know, because it's very defined, and there's always question marks over whether that media is aberrant in some way. But we've got to move towards that, and the immune compartment could well be a good tissue, if you like, to look at in that regard. What we have in my lab is we've got a rule of thumb. We see effects on cells in culture, and we might start, we work on PKM2, for instance. We always confirm in vivo to make sure that what we're seeing is real, just for fear of an artifact, really, you know. But I do think it's feasible, absolutely, as a possible model system in many ways, especially as there are these real differences between different immune cells.
Thank you.
So Jennifer, really quick.
Yeah, I have the exact reverse of what Vamsi was asking. I'm interested in how the immune system, these cells, which are circulating continuously throughout the body, intermixing with different types of tissues. How do we get at that type of intercommunication? To what extent do you see the tissue itself as the key integrator for what these immune cells are doing?
It's also extremely important, I have no doubt about it, because hypoxia is really important, obviously. So the immune cell goes often into a hypoxic environment, actually, you know.
Yeah.
Or in a very acidic environment. I mean, many inflamed sites are very acidic. The pH in the rheumatoid joint could be as low as 3 or 4, you see. And then the macrophage goes in there, has to respond to that environment. So again, it's a good example of a real life situation there in many ways. I'd love good, now there are, as you know, there are good imaging technologies out there for hypoxia and acidic environments. They'd be very useful to integrate here. And we know the cell will change its behavior in that environment. And then very importantly, the macrophage, say, will talk to the fibroblast. Right, and drop it into a different state as well, you see. So that's a really good aspect of this as well.
There are a few questions from the audience, and one of them asks, how does the itaconate work on different immune cells in different tissues?
Well, it's mainly made by, it's a strange metabolite. I could have spoken all day about that, right? It's my favorite thing, but it comes off Krebs from a connotate. It's only really myeloid cells make it. So macrophages are a great source, for instance. The enzyme, it's ACOD, isn't it? It's kind of decarboxylase. It's only really in macrophages, but it does get released, and you can detect it in millimolar concentrations, just cellularly, you see. So the question then becomes, is that acting as a signal Can other immune cells take it up, you see, and then it gets inside? There's probably a receptor like there is for succinate. So that's one of the metabolites we use as evidence for a cytokine-like profile here. Now, some of that is still a work in progress. Again, getting back to Vamsi's, it could be a bit artifactual. The concentration is very high. So we're starting to work that one out as to what it might do as an extracellular signal, you see. We're still trying to work on that.
Another question asked, does the population...
Wrong, I'm sorry. I think we need to move on to the next speaker. Let's try to circle back to these questions maybe later if there's extra time.
Okay, let's do that. So thanks again, Luke. The next speaker is Joshua Rabinowitz from Princeton University. Josh, please.
Hi, it's fantastic to have the opportunity to follow Luke's talk and to speak in such an innovative meeting. And I'm going to talk about metabolic flux at the whole body level, but I will try to get us back to concepts of single cells and heterogeneity as I go along. And so ultimately, my lab is interested in two questions. What are the fluxes across 1 million organs and cell types? And how are these fluxes controlled? And these are big questions that definitely need involvement of the community to answer them. And we think about these questions within a framework of mass conservation, or equivalently flux balance. Fundamentally, what makes metabolism at some level simple is that whatever goes in must either come out or be stored. And this is a very special constraint that applies to metabolism and not most other biological systems, and makes it tractable. And so this applies to the whole body level, it applies to every cell in the body, and it applies to communication between cells and organs in the body, because ultimately one cell's waste, if it's not excreted from the body, needs to be another cell's fuel. Now we're interested in about 1,000 fluxes in every cell type in the body. How can we begin to figure out what these are? Well, we have a few measurable things. The classic thing to measure is arterial venous gradients. And with metabolomics, this is powerful, but it's also coarse because it measures what's net made in each vascular bed. And think about the vascular bed of the leg, okay? It has skin, it has fat, it has bone, it has multiple types of muscle, right? So we need to get more granular, and we can get more granular, as we've heard, with isotope labeling. Okay? You can do this looking just in the bloodstream, and that tells you whole body production and consumption of metabolites, but then you can follow that downstream. And so when we follow downstream, we can start by saying what's big at the whole body level, what's the biggest fluxes? And so the biggest one is lactate. And so that's really interesting and surprising. And people have known that since the 1970s, but the thing that really made, I think, us believe that lactate is more than a waste, but actually a fuel, is the fact that when we trace from infusion of lactate into the TCA cycle, we see that lactate labels the TCA cycle in every organ in the body. And in fact, when we do this in a quantitative way from glucose and from lactate, we see lactate labels more, and that the glucose contribution can be explained by the fact that glucose labels circulating lactate. That is the main path from ingested carbohydrate into the TCA cycle, goes through circulating lactate. Now you heard from Tara, beautiful work on trying to figure out where this lactate is made. I want to say a bit about how its consumption is regulated. And we're excited that we have now found a key regulator of circulating lactate consumption, and that regulator is insulin. When we apply insulin, we roughly double the contribution of circulating lactate to the TCA cycle. And this, we think, occurs through the fact that insulin suppresses lipolysis. And so circulating fat drops, and we can completely suppress this effect of insulin on lactate use by infusing lipids to keep lipids at the normal pre-insulin level. Okay, so ultimately, glycolysis in the TCA cycle can operate independently, connected by circulating lactate, whose consumption is promoted by insulin. Can we begin to get granularity spatially to these measurements? Well, you heard a little bit about this from Tara. I'll just show a couple of additional examples. One additional example is that when we look in the kidney, we can distinguish part of the kidney that's glycolytic, the center, or medulla of the kidney from part of the kidney that's gluconeogenic, the cortex. And this is very clear, okay? And it's a beautiful, vivid illustration. Can we learn things about how diet interacts with metabolism in this way? Right. On a normal diet, if we look at the brain is homogeneously labeled in the TCA cycle from glucose. Everywhere in the brain, glucose is the dominant TCA fuel. But if we switch from a normal carbohydrate-rich diet to a ketogenic diet, what you see is, of course, ketone bodies start being used. But what's interesting is they aren't used homogeneously in the brain. There are parts of the brain like the hippocampus that are avid ketone users. There are parts like the midbrain that remain almost purely glucose using. So we have a lot of work to understand how this is controlled and why it matters to brain physiology, but this is clearly programmed into the brain. The different parts have strong glucose versus ketone preferences. So ultimately, to go further, we need to improve the spatial resolution of imaging mass spec. We need to build methods for integrative modeling where we combine AV data, the actions of each organs into whole body models. And we need to move also from thinking about fluxes as simply contributions, like how much of TCA carbon comes from glucose versus ketone bodies, to thinking about absolute flows, because you can have changes in fractional contributions, but that doesn't tell you how fast the whole system is running. So I want to wrap up with some examples where we get into how fast is the whole system running and how can that impact how we think about biology. And so to get at this, we decided we really needed to bring in this 4D, the dynamic component. And so we infused lactate, but now in a very rapid way, where we have a minimally perturbative, but almost instantaneous way of lasing lactate labeling in the bloodstream. And then we're going to look at how fast does the TCA cycle label downstream. And this is all possible because lactate is taken into cells and taken into mitochondria so fast that we can use it as a probe of overall TCA turning rate. On the left, you see that within 30 seconds, we can raise lactate labeling in the bloodstream to a steady state labeling value. And then we can look downstream in different muscle types and see, for example, that in diaphragm, which is working, which is a red muscle with a lot of mitochondria, it's just whirling its TCA cycle. And in quadriceps, a white muscle that's not working as hard, it's going very slowly. So we see dramatic differences. Ultimately, we can convert these labeling rate constants by multiplying by the pool size of TCA intermediates into absolute fluxes. We can add them up across the body. And as a sanity check, when we add up the NADH production in the TCA and associated oxygen consumption, we account quite precisely for whole body oxygen uptake. Now, where we learned something really surprising and interesting with this was going into tumors, as Luke alluded to. And tumors, have a yin and yang history in terms of TCA usage. If you really believe Warburg hardcore, you might think tumors don't really need their TCA. But in the past decade, we've heard about how tumors actively select for healthy mitochondria. How in beautiful work from Ralph DeBerardinis, he sees that lung cancer actually puts more glucose carbon, fractionally speaking, into TCA than normal tissues lung does. But when we now bring the dynamic, the fourth dimension into this and look at absolute turning rates, what you see is that tumors clearly turn their TCA cycle more slowly and by a big amount. They also have lower TCA intermediates. And this results in somewhere from 5 to 10-fold decrements in TCA flux in tumors compared to the tissue of origin. And what this means when we integrate it with quantitative measures of glycolytic flux is that tumors, because they have elevated glycolysis, are making more ATP glycolytically, but they're still making a majority of their ATP in the TCA cycle, and their whole ATP production rates are down 3 to 10-fold compared to the tissues of origin. We have this now in mouse models for the 3 biggest cancer killers: lung, pancreatic, and colon. So this is a really new view of tumors, I think. Starting with metabolism, the tumors may actually be hypometabolic. They may meet their anabolic needs, not so much by upregulating metabolism, but by turning off basal ATP using functions. So ultimately, metabolism is the oldest part of biochemistry. and most enzymes are known, but we have a lot of work to do to figure out how do cells work together in vivo? How does this change with diet and exercise, as Vamsi alluded to? These are the biggest health promoters. And how can we translate this knowledge into personalized dietary guidance? So with that, I'll stop and take questions, and thank you all.
Fantastic. And I already see hands up, Jared.
Yeah, Josh, that was provocative and elegant as usual. So I wanted to ask you about the TCA cycloflex measurement in tumors. I think that's really interesting and fantastic. Does that just measure TCA cycle flux from lactate, or can you essentially infer that that is TCA cycle flux regardless of substrate because of the nature of the turnover rate?
Yeah, so these are dynamic measurements that we think are invariant for whatever the substrate is. We have the exact same results using glutamine instead of lactate as the probe for pancreas because it uses glutamine a lot. Glutamine is a less universal probe of TCA dynamics, but we think this is a fundamental measure of TCA turning rate.
Cool.
And a question from the audience. Can you please elaborate insulin regulation of glucose as compared to lactate? Is it possible insulin could regulate metabolism of other carbon substrate as well?
Yeah, I'm sure that insulin regulates many things. I think it's striking to us that we think so many of the actions of insulin are downstream of its effects on adipose and lipolysis. And we do think that a key component of how insulin regulates glucose homeostasis is by promoting circulating lactate clearance. And then there's crosstalk from the lactate to glycolysis. We're still working on trying to figure out that crosstalk, but we're have pretty good evidence that it exists.
Okay, Ralph, go ahead.
Josh, excellent. I mean, adding the quantitative component to this is so important. Do you have any insight into what turns down the ATP consumption in the tumors?
In pancreatic, we do have some insight. So pancreas, exocrine pancreas just makes a ton of digestive enzymes normally. And so it has the highest protein synthesis rates in the body. And we see the protein synthesis is markedly suppressed in pancreatic cancer compared to normal pancreas. So we think that's the case for pancreas. I'm sure it's a different answer for each cell type or tissue of origin, depending on where that tissue of origin normally spends most of its ATP.
And if I could ask one quick follow-up really quick, what are the measurements you would have to be able to make to do this in patients?
I know this is a hard one. So this is based on dynamic sampling from the TCA cycle. So you have to have serial biopsies, but I don't think that would necessarily be too bad in patients because you could have serial needle biopsies.
Yeah.
You have to give that lactate much faster, but the amount of tracer you need is not much because the whole thing is so quick.
Right.
So there's a bunch of questions from audience on the same topic and about how the tumor cells turn down ATP production. And one of them asked, do you know that when the ATP production is turned down and other metabolites production goes up?
I think the short answer is we actually don't really know that. I mean, we know that lactate production goes up, right? That's the Warburg effect, and that certainly is the major fate of the glucose that's consumed. But I think we don't have, deep measurements of, shunting of this in anabolic pathways, other than the fact that the classical things happen like nucleotide synthesis. But there's not nearly enough nucleotide synthesis to account for the extent of these other changes.
Okay, I still have two questions from the panelists, and so Mark first and then Lou. Mark, you're muted.
Say, Josh, it was a terrific talk. I really enjoyed it. But I'm wondering if this stable effect in tumors versus non-tumor cells, if it was really stable, you should see it, or you might expect to see it at the level of protein expression, at the level of protein modification, even at the level of transcription. So looking back at this, these are not the genes that cancer people usually looked at first, but I'd be wondering if that state that you're describing, or the difference in states you're describing, is seen at those levels as well?
It's a great question, Mark. And I think we haven't done that analysis well enough yet, but I think it's an analysis that we should prioritize. I mean, I think one of the most interesting questions in terms of metabolic control is how much of it is through the things you're alluding to, the expression and modification of enzymes, and how much is driven by either substrate availability or demand. And so I have a sense the origin of this effect may be in turning down ATP demand, but it's probably an intersection of both, and we should look carefully for that.
Lou, please.
Sure. So Josh, first of all, that was a terrific presentation. I just want to make a comment that you've really, I think, your data really very accurately explains the Warburg effect. The problem that Warburg had and his followers, F. Racker in particular, was why does the ATP drop if you're doing, more glycolysis? Why isn't ATP levels higher? And you point out that even though pyruvate's going into the mitochondria, that you're not getting ATP produced in the mitochondria either. So that's why they thought there were defective mitochondria. But almost certainly the explanation that I'm sure you would make, that I would too, is that it's all because the mitochondria are making citrate and using that for fatty acid synthesis in order for the tumors to grow. And the other point is that you have to drop the ATP if you're going to do glycolysis at a high rate, because the first step in glycolysis, phosphofructokinase, is inhibited very acutely by ATP levels. So dropping the ATP and doing fatty acid synthesis explains the whole Warburg effect. And I think your data really is very beautiful on that.
Okay, great discussion. Thank you, everyone. Thank you, Josh. Let's move to the last talk of this session, will be given by Ashley Solmonson from UT Southwestern. Ashley, are you ready?
Yes. Let me share my screen here. Oh, this is not letting me share my screen. Please hold, sorry.
There must be top secret in your talk. Your computer
Yeah, I guess. I'm so sorry about this. Let's see. There we go. Oh, I'm really sorry. This is giving me a hard time. Let me take a minute.
While we're waiting for her, if anyone has questions, we can bring it up for discussion. I think Mon has one that I cut her off. Go ahead, Mon.
Yeah, so it's beautiful talk, Josh. So I just wonder, considering the metabolic adaptation, if you do short-term versus long-term keto diet, whether the brain will change the preference to use keto differently.
Yeah, we haven't explored that yet. I think it's a great question, and it would be beautiful if we saw spatial shifts in ketone usage over time as people get deeper into ketogenic diets. So great idea for an experiment. Thank you.
Thank you.
All right, looks like Ashley is ready. Go ahead.
So sorry about that. All right. Thanks to the organizers for selecting my abstract to be able to talk. I think my work fits in really, really nicely with this workshop. So I'm excited for comments. So today I'll talk about metabolic transitions during mammalian development. So I work in Ralph DeBerardinis' lab. I'm a senior postdoc in the genetic and metabolic disease program. And we study inborn errors of metabolism. And one of the issues of inborn errors that's been an issue has been this idea of tissue specificity. So for individuals who say have mutations that impact mitochondrial function, you also see these diseases manifest in some tissues, but not all tissues of the body, despite the fact that almost all the tissues in the body require mitochondrial function at some point during the lifespan. And I'm specifically interested during development because most inborn errors don't actually present symptoms until the postnatal period. And so this begs the question, does tissue specificity occur in utero? And can the placenta, which is the ultimate transient organ, and is derived from fetal cells, so shares the same genotype. Can the placenta compensate for deficiencies of the fetus, or can maternal tissues compensate for deficiencies of the fetus, such that when those are removed, these symptoms manifest and the disease progresses? And so in terms of looking at development, we chose to focus on mid-gestation. Today, I'll talk about that from the standpoint of 9 days after conception to about 12 days after conception. This is post-implantation when vascular agenesis is increasing in both the embryonic side and the uterine side. And this increases oxygen availability, which also increases nutrient delivery and ultimately facilitates rapid growth. It also plays a role in hypoxia during development, which is known to regulate cell fate and other aspects of developmental events. And this is also happening at the beginning of stages of organogenesis, where the heart and liver and brain are among the first organs to form. And we know from the literature that many mouse models in which mitochondrial activity is perturbed, these mouse models often result in embryonic lethality right in this mid-gestation range. And it's been suggested based on the oxygen changes and the mitochondrial function that this is a period of an embryonic shift in which the embryo goes from a low oxidative phosphorylation state to a higher oxidative phosphorylation state. But this hasn't actually been shown in vivo per se. So we kind of set out to ask these questions and ask general questions about metabolic transitions that might occur during development. So we started from a very basic approach to just take whole embryos and whole placentas, including the decidua, and look at the metabolome over time in mid-gestation from day 10.5 to 13.5. And you can see in the PCA plot that both embryos and placentas have very specific metabolic profiles. That's probably not so surprising. But I think what was surprising is how different the metabolic profiles are day by day. And the heat map on the right is showing you that that can happen at the level of individual metabolites or individual pathways. But what the clustering at the top of the heat map also shows you is that in both tissues, day 10.5 is an outlier from the rest of the days that we looked at in our mid-gestation series, suggesting that from 10.5 to 11.5 days post-conception, a metabolic transition occurs. It occurs in both the placenta and the embryo, and it's different in both tissues. And so we're really interested in what might be driving that metabolic transition and how different tissue types respond to that metabolic transition. So our lab is well known for doing stable isotope tracing in vivo, and I did some optimization to apply this to the developmental side of things. And specifically, we can take advantage of this aspect of mammalian or mouse biology, and that most pregnant mice have between 5 and 10 embryos at a time. So we can do an experiment that I'm calling a serial C-section, in which we place a catheter in the tail vein of an anesthetized mouse. We make an incision to expose the uterus, and then we can isolate out a single concepsus, that is the placenta and its matched embryo. And at the same time, if we take a retroorbital blood bleed from the mother, we can get a snapshot of our infusions and see how dynamically the tracer is incorporated into each tissue, and then also how it's oxidized downstream. And so we've done this with uniformly labeled glucose on the left. Glucose is a known major nutrient for developmental tissues, so you can see that the tracer reaches an equilibrium at all 3 compartments very quickly. And this is different to what we see when we infuse glutamine. So glutamine reaches a steady state when we do this infusion in the blood, and a lower steady state in the placenta, and it never quite reaches the steady state in the embryo. And so these are things we have to consider not only in terms of transport of these nutrients across the compartments, the placenta and the embryonic compartments, but also we have to consider those when we think about the downstream oxidation of those nutrients as well. So we wanted to test whether this embryonic shift was occurring. And so we switched over to steady state infusion experiments with uniformly labeled glucose. And we asked in a 9.5-day embryo, relative to the amount of glucose that gets in the tissue, how much of that glucose is getting into the TCA cycle in the placenta versus the embryo. And you can see at 9.5 days, the placenta seems to incorporate a little bit more glucose in TCA cycle metabolites compared to the embryo. However, if we look at subsequent days in mid-gestation, we see that with each day passing, the embryo starts to incorporate more and more glucose-derived carbon into the TCA cycle, suggesting that this oxidative shift or the embryonic shift that has been proposed is likely happening to some degree. The problem with this is that the embryo is not just developing as a single cell type that's growing rapidly, it's also undergoing differentiation and the beginning stages of organogenesis. And so we needed to look and see if this was playing a role in different cell types and different tissue types as they're developing in the embryo as well. So we moved over to 12.5 days when we could reliably dissect out individual fetal tissues. And using the uniformly labeled glucose infusions, we were able to isolate brain, heart, and liver and show that brain and heart are slightly more glucose avid than the liver, incorporating more glucose into TCA cycle intermediates relative to the glucose that got into that tissue. And this is not to say that the liver is not oxidative, because when we do an infusion with glutamine at the same time, we can see that the enrichment from glutamine is higher in TCA cycle intermediates in the developing liver comparative to the brain and heart. So this tells us that by 12.5 days during developmental organogenesis, metabolic preferences are largely set and may be dynamic over the course of the organogenesis process. So I mentioned that we are interested in inborn errors of metabolism. And so we wanted to know if our methods were sensitive enough to be able to identify changes caused by inborn errors of metabolism. And we've previously reported on this LIPT1 deficiency that was identified in a patient from our clinic. The patient had a hypomorphic allele, this N44S mutation that we knocked into the mouse LIPT1 locus. LIPT1 is a nuclear encoded enzyme that goes to the mitochondria and transfers a lipoic acid cofactor, which is essential for the activity of 2-ketoacid dehydrogenases in the mitochondria, like pyruvate dehydrogenase, branch chain ketoacid dehydrogenase, and alpha ketoglutarate dehydrogenase. And we were surprised to find that when we knocked in this patient-derived mutation in the homozygous state, this resulted in an embryonic lethality from 10.5 to 11.5 days. And so we use this model to ask, can we identify LIPT1 mutant embryos based on their metabolic signature alone? And the short answer to that is yes. So the graph on the top, I'm showing you a uniformly labeled glucose steady state infusion in a heterozygous dam. And we can see that the homozygous mutant embryos shown in red incorporate less glucose carbon into TCA cycle intermediates than the littermates that are in utero at the exact same time. And then the fact that the placenta is derived from fetal tissues and shares the genotype of the embryo, we expected that there might also be a defect in the placenta. And we do see some perturbation of glucose metabolism in the placenta, but not to the same degree that we see in the embryo, suggesting that there are differences in how the embryo and the placenta respond to LIPT1 deficiency. And ultimately, we were interested in what was causing the lethality of our LIPT1 mutant embryos. And so we started looking at placentation and placental function, because this is an important aspect during mid-gestation. And we can do this by using our tracing ratios to try to understand different aspects of this. So if we take the ratio of the tracer in the placenta relative to what's in maternal blood, this gives us a measure of placental glucose uptake. Moreover, if we take the ratio of the tracer glucose in the embryo relative to its own placenta, this gives us a measure of placental glucose transfer from the placenta to the embryo. Neither of these were altered in our LIPT1 mutant tissues, nor were they altered when we infused glutamine, which suggested to us that the placental function of transporting nutrients was intact in the face of LIPT1 deficiency. And we also looked at placental differentiation markers, and none of these were really robustly changed, suggesting that our LIPT1 mutant tissues, our LIPT1 mutants did not show a placental defect, which was ultimately leading to their lethality. We looked at a lot of other things, but one of the things that was quite interesting that we hit on was during the period of mid-gestation, erythropoiesis needs to occur because the embryo is getting larger and it's getting large beyond the capacity for oxygen diffusion. So erythropoiesis has to occur, and we can identify terminally differentiated erythrocytes in the fetal context by looking at CD71 positive, TER119 positive cells in the whole embryo. And we see that these were markedly decreased in our mutant embryos compared to healthy litter mates. And when we looked at cells that had CD41 positivity, which were indicative of megakaryocyte and erythroid progenitors of those erythrocytes, we saw that those were largely elevated, suggesting that there's a blockade in the differentiation of erythrocytes due to LIPT1 deficiency in this mouse model. And this was quite surprising, and we were really interested to see if this matched up with our human model or our human patient where the mutation ultimately came from. And looking back at the chart, we find that our patient is actually chronically anemic from a very young age, and that this effect of low red blood cells is really only consistent to the red blood cells. It's not affecting the white blood cell count or the platelet count to a similar degree. That suggests that maybe what we've identified in the mouse model is consistent with some aspects of the human disease and gives us an opportunity to get a handle on other aspects of this LIPT1 deficiency disease and how we might potentially be able to treat it. So just finally, I want to summarize and say that here I've told you that we've identified a metabolic transition that occurs during development between gestational days 10.5 to 11.5. We find that there is an embryonic shift that occurs with the embryo increasing glucose oxidation in the mitochondria over mid-gestation, but that mid-gestation fetal tissues have very specific metabolic fuel preferences. And ultimately too, that LIPT1 activity is required for erythropoiesis. And so this idea of in utero tissue specificity when it comes to inborn errors in metabolism might in fact be correct and should be something that we think about when we think about the context of inborn errors of metabolism. So with that, I'd like to thank Ralph. He's been an excellent mentor, letting me go down this weird development route that none of us were familiar with. We have excellent collaborators, and our CRI Metabolism Core has been wonderful. And I'd like to thank my funding as well, and I'll take any questions.
Thank you, Ashley. That's a fantastic talk. So I see hands already. Daniel, please unmute yourself.
All right. Ashley, great presentation. I loved it. I have a question related to the hormones that the placenta produce, like HCG in humans and progesterone. These two are different in that one has high glycosylation, so it requires sugars, and the other one is cholesterol based, which is lipids, right? So you're seeing this glucose shift to oxphos utilization, right? So do you know if any of these hormones contributes to the shift you are studying?
Yeah, so we haven't paid much attention to the hormones at this point because we have focused a little bit more on the embryo in this regard to try to understand the differences. But I can tell you that there are distinct differences in how the placenta metabolizes glucose and how the embryo metabolizes glucose. The increase in glucose oxidation that we see in the embryo doesn't happen in the placenta. The placenta basically remains flat in terms of its glucose contribution into TCA cycle intermediates. And there are also differences in the rate at which the placenta generates glucose 6-phosphate, maybe not the rate, but we see a dilution at the level of glucose 6-phosphate in the placenta that we do not see in the embryo, which suggests that there are differences in glycolysis and carbohydrate metabolism between the placenta and the embryo. And it may play a role due to hormones that are either produced by the placenta or in response to hormones in the embryo. So yeah, that's a good question.
And there's a question from the audience. I wonder how maternal-fetal interaction goes in the fetal metabolic adaptation maturation process.
Yeah, these are really, really interesting questions. I think we've just, in looking at the literature, there hasn't been a lot done on developmental metabolism in this sort of way since maybe the 80s, 70s, 80s, and they were largely done on large animals. And now we have all the tools we need to disrupt metabolism in all three different compartments, maternal compartments, the placenta, and the embryo, to really understand what is the cooperative nature of these compartments during development, and how does that change over development once the embryo, say, has the ability to generate and synthesize insulin and other factors like that. So these are really important questions.
So one more question here from the audience, and asking about the mitochondrial reprogramming driving erythrocytosis, and what do you think the connection is?
Yeah, that's an excellent question. So I think right now our focus on why LIPT1 deficiency might lead to disruption and differentiation of erythrocytes. We know that there are mitochondrial metabolites like succinate, that are succinyl-CoA, that are required for heme biosynthesis. Heme is important and transcription and of different genes during hematopoiesis and erythropoiesis. So that's one option. We also know from some of our experiments in the fetal system, in the fetal tissues, that the liver really likes glutamine, and OGDH is really important in terms of glutamine oxidation. And so maybe LIPT1 disrupting OGDH activity could disrupt glutamine oxidation, and maybe also disrupt the alpha ketoglutarate succinate levels. So there are a lot of options for mechanism, and we're interested to dive into those and see if they hold true, to get a little bit more information about why this inborn error is causing a deficiency in one specific cell type, as opposed to many others that also require mitochondria.
Excellent. So I think our time should move into the panel discussion now. And so now we can bring questions up for all panelists. And I think we have a diverse group of presentations, but we all centered around the metabolic specialization. Looks like different cell types have different metabolism. And so I wonder if the panelists could comment on how this is regulated, and how the different cells with different type of metabolism could potentially interact in an in vivo situation. Should I call Luke?
I'll say Wrong. Well, for me, it's
sorry.
Remember, signaling pathways coming off immune receptors are changing metabolism. So we mapped a pathway from TLR4 to mitochondrial change to succinate to HIF, and then HIF drives a set of cytokines. So in other words, we kind of see them almost as signaling processes in a sense. And then we're trying to get a handle on how these receptors actually engage with signaling pathways. And things like mTOR and AMPK are very important as too. I mean, immunologists have worked on those signals for years, remember. And they are metabolic sensors, mTOR and AMPK. So in other words, we've been dabbling in the area of metabolites and signaling for a long time in immunology. And we need to find out more about the wiring of these pathways dynamically during activation with specific signaling pathways.
Josh?
Yeah, that was a great lead in, Luke, because I always think about it from almost the back end, whereas Luke thinks about it from the front end. And from the back end, when you think about these metabolic flux changes, ultimately they're programmed either in metabolite concentrations or enzyme concentrations. And we know for microbes that the biggest driver, at least in our experience, is environmental nutrient concentrations. And this is the thing that is the strongest determinant. But in vivo, we obviously have metabolic homeostasis. And so then it comes down to, what are the impact of diet on circulating metabolites, and what's the geometrical position of these cells relative to vasculature, as we heard about in the liver, that's determining kind of metabolite access. What's the demand side? What's the tissue needing to make that's consuming metabolites? And then that has to all be integrated within the context of what enzymes are expressed and where they are, of course, in the cell.
If I can comment on that from the developmental perspective, I think from a developmental world, we know that signaling molecules like Wnt and BMP play a role in patterning And those are really important. And we know that there are metabolic gradients formed, as Josh said, by the expression of metabolic enzymes. But what we don't know in development is if individual metabolites can act as morphogens in the same way that signaling like Wnt and BMP do. And I think that that would be really interesting in regards of development, if we can pick up on some of those aspects as well.
Okay, I see questions from Heidi and then Daniel.
Okay, I'm unmuted. Sorry. Thank you very much. And I wanted to touch on something that Luke was saying about the sort of evolutionary aspect of these inflammatory switches and things that can change. I mean, we've been doing some work in my lab about the role of mitochondria in inflammation and our Parkinson's projects and things like this. And, you know, we see a really rapid response of the generation of mitochondrial vesicles in the first few minutes. And the way I think about it in evolutionary terms is often like, you know, when they were bacteria, if they get it hit with phage, the bacteria will shed vesicles and they change their metabolism as well. And so do you ever think about, you know, that level of integration and how it might have evolved in terms of the transition from innate to adaptive immunity through evolution, and how mitochondria might play roles? Because you describe it as, you know, the enemy from within. But I think these signals mitochondria are sending are signals that have co-evolved to protect the host. But I don't know what you think of what actual immunologists think about that.
That's a good philosophical question, Heidi, in a sense, Heidi. I mean, I often think of it as they're sitting there like bacteria, let's face it, you know, there's a homeostatic state, isn't there? And we're tolerating the mitochondria, and there's kind of a literally an endosymbiotic relationship between the mitochondria and the host cell. But it always strikes me as interesting that gram-negative bacteria are great at changing mitochondria, you know? So it's almost like if they're calling back to their old friends in a way, you know, and then the mitochondria begins to release its DNA as you know, a great example. So
yeah,
that DNA is being sensed as if it's bacterial DNA, and now there's sensors for that.
Well, Jerry Shadle would say that the mitochondrial DNA release is really something that is used as a signaling molecule to inform the nucleus to change, right? So it's not necessarily a bad thing, you know, these things.
No, I think this is happening all the time in a homeostatic way, and we need it, you know, and the big question is, when does it pivot into a damaging event, maybe, you know? And then you're drawn into reactive oxygen, and you're drawn into antioxidant processes to contain it, you know? But it's a really interesting question, isn't it? And we know that in Th17 cells, they don't really use OXPHOS much, it's mainly glycolytics, whereas the regulatory T cell has a very coupled mitochondria. They're really healthy, you know, they're respiring normally. So there's something funny going on. Some view it as a rapidity of response. In other words, if you have an inflammatory process, an infection, you need to act quickly. So you ramp up glycolysis rapidly to make loads of ATP to develop, and a bit of biogenesis as well. You see, it takes too long for mitochondrial biogenesis to happen. You see, that's one view. It could be a time-dependent thing, maybe, you know.
Yeah.
But good questions.
I mean, your signaling switch from IL-1 beta to IL-10 is just astounding, and I think there's so much exciting stuff to study there. So thank you very much.
Isn't there? Yeah. And that was a weird experiment that Mike Murphy recommended. He said try as SDH inhibitor, and we did, and we got that interesting result.
Mike knows everything.
Yes.
So to follow up this point, and there's also a question from the audience, and asking, you know, how much do we know about what drives the change of metabolic flux? And the point is, the technology has developed so that we know a lot of metabolic reprogramming, but what's the driver of the reprogramming?
Well, it's gene expression, Rong, because we showed that in 2013. You get a big increase in expression of certain metabolic enzymes. You know, they're turned on, and then you begin to change the flux simply through increasing the levels of certain enzymes in these pathways became clear, you know. And then someone mentioned the last session, these nutrient transporters are critical, remember, because if they go up, you're going to bring in a different set of nutrients of the cell, and that will feed into these pathways as well. Hence and we all know this, the multi-omic approach is important, because if you can correlate gene expression change with metabolic change, then you're onto something and trying to explain the dynamics of these pathways, I think.
Yeah. And I'll just add the beautiful work of Adam Hughes in terms of the removal of mitochondrial nutrient transporters dependent on amino acid levels through this MDC compartments. These are rapid events that happen quite quickly. So it's you can totally change metabolism by altering that. And so Jared was really picking up on a cool thing. So.
Yeah.
Okay, and so we have a question from Danielle and then Mon.
Yeah, so this question is for Joshua, and it's related, going back to tumors and reduction of ATP consumption. Would it be possible that tumors generate metabolites that are perceived by the surrounding, and these cells are then the ones that provides the ATP, and then those cancer cells can consume the ATP from other cells in the surrounding? Is that possible?
It's certainly a cool idea. It goes back to the metabolite transporters that we were just talking about. I don't know about adequate transporters to do that ATP transfer, but I'll tell you, although I'm incredibly skeptical of anyone who ever calls any phosphorylated metabolite a substrate, we have data that I don't understand on one extracellular phosphorylated metabolite recently that totally has surprised us. We're still trying to work through it. So maybe there are big surprises like that, where some extracellular ATP can be used to drive intracellular ATP in novel ways. So a cool thing to dream about, definitely.
Yeah, I have a question for Luke. So I think it's really fascinating, the modification of proteins by those different metabolites. So I think it's kind of one way how metabolite interact with micromolecules. Like Gerard, I think, also mentioned that. I think, it's amazing, like we keep saying more and more of them, but if we think about, for example, histone modification, people talk about you need a writer, you need an eraser, you need a reader. So how do you think, those modifications that from metabolite, how they got there and how they're removed and how they function, to regulate things, and how can we look at them more, I think, systematically?
I think it depends on your biases in a way. Like if you believe phosphorylation and dephosphorylation was the first, let's face it, and that's the parent of all this in some ways, and then acetylation and deacetylation. There's always the yin and the yang, and it's no surprise then that succinate and alpha Kg are behaving in that way as cofactors, as you probably know, for demethylases, and they're coming off Krebs, for instance, to regulate demethylases. There's loads of examples like that. What gets me is all these new ones. Do they mean anything at all? I don't know. I mean, you can measure them. I'd love to know what they mean. I think that they may not mean anything. That's a bold statement in a way. We don't think you can. Like succination is a good example with fumarate. That does seem to be an irreversible event. It doesn't seem to be an enzyme to change that modification back to normal. Itaconate the same, but that's an alkylation reaction. We haven't found a way to take itaconate off proteins yet, so maybe irreversible. And then there's ubiquitin is in the mix as well, you see. So I think we're just beginning to try to address those very questions. And the point I made, like there's such diversity in the proteome because of the covalent modification. We've known this anyway for decades, really. But it turns out the early observations were the tip of the iceberg. And who knows where that's going to go next. And I'd love to get more on that, really, I mean, I don't know what the number is. There could be as many as 30 modifications coming off metabolites. What does it all mean in terms of function of those proteins ultimately? And we're just beginning. We know it means things like degradation or enzyme activation, phosphorylation again being the initiation of this. But we'd love to get more of a handle on what these outputs mean. They hopefully will mean something. They'll add to the complexity of the phenotype for definite, because it's such an intensely complex thing. So I'd love to get more of a handle on that way.
So we have a few minutes left, and I want to circle back to this question that came up in several sessions, which is whether the metabolism is a driver of cell function or whether it's just a requirement for cell function at different stage. I think all the speakers in this panel touched this question, so maybe you can all give us your thoughts on that.
That's a chicken and egg question, Rong. Chicken and egg, you see, is the problem. That's my first response, right? We know if we use pharmacological intervention, you can change the fate of a cell. So therefore, that would suggest the metabolic change is determining for the fate of that cell. But equally, the fate will change metabolic profiles as well, you see. So I think it's a difficult one to answer. As I said earlier, you can phenotype a cell based on its metabolism. Does that mean that the metabolic change is governing the phenotype? I think it probably does mean that,
Yeah, I want to insert here that years ago, we're trying to address this question. So we've generated some transgenic mutant models that would model certain state of metabolism, which is not normally observed. And we thought that that would drive the disease phenotype. Actually, it didn't. And you know, we've generated mice that has an inflammatory metabolism phenotype. It doesn't automatically inflame, and it needs another stimuli, and then it could either exacerbate or reduce the response. So if I think metabolism as a driver, I would say, you know, if it's a bad driver, it won't cause accident unless there's gas in the car. So maybe we should think it in a more sophisticated, complex situations for that.
Yeah,
it's a modulator, I guess you'd say.
Yeah, if I can kind of jump in here too, I think it also depends on when these events are occurring. If we think about 4D, right? If you take cancer metabolism, for instance, I think the initial stages of driver mutations changing metabolism is the genetics changing the metabolism. But at later stages, you know, when you have a hypoxic tumor or, you know, different effects like that, then I think there are ways in which the metabolism feeds back and changes the mutational status and can influence different aspects like that. So I think looking at when the metabolism is acting really plays a role in that chicken or egg question.
I'll just say that metabolism is flexible, and so it's very responsive to supply and demand. A lot has to go wrong before you get something like ATP depletion. So from that perspective, it does have this kind of housekeeping function. It was always ascribed. At the same time, the ultimate proof that metabolism is a major driver is the fact that diet impacts health. And it probably, you know, at the population level is the #1 driver of public health right now that can be improved. And so I think that's the proof that metabolism is in the health driver's seat.
If I may, just going back to my earlier example, there kind of isn't a more clear example of metabolism being a driver than the pancreatic islet. Glucose metabolism generates the signals that drive insulin secretion. And I think we may have other examples of that kind of innate signaling from metabolism that we have yet to discover.
All right, and any final thoughts on this session? If not, thank you all very much. And it's been a really lively discussion and very stimulating. And so I send this back to Jeannie, and we're actually on time.
Yes, perfect. Thank you, Rong. Thank you, everybody. That was a great session. Let's take another 5-minute break, and we will resume at about 3:27 p.m. Eastern.